It is time. Good afternoon, good morning, good day, wherever part of the globe you are. It is our pleasure today on behalf of Dr. Hanley and myself, we are serving as the co-PIs of the coordinating research on emerging arboviral threats encompasses the neotropics, or CREATE-NEO, who is the sponsor of this seminar series, to welcome the first seminar series for this calendar year. We are very pleased that you are able to join us. Before Dr. Hanley introduces our speaker for today, I would like to let you know that next month we will have Dr. Sandra Verges Lopez from Gorgas, Panama, presenting. So we are looking forward also to see you there. A couple of housekeeping notes. If you have any questions, either please put them in the chat or raise your hand, and Dr. Hanley or myself would be able to facilitate your questions and so forth. I will stop here, and Kathy, please.
Well, it's my pleasure to introduce our CREATE-NEO collaborator, Dr. Daniel Becker. Dan received his undergraduate degree in anthropology, global health science, and society from Bard College in New York. He then did his PhD at the Odum School of Ecology at the University of Georgia. He did postdoctoral fellowships at Montana State University and Indiana University. And he is currently an assistant professor in the Department of Biology at the University of Oklahoma in beautiful Norman, Oklahoma. And Dan has spent much of his career working on bats and bat-borne diseases, and I'm sure some of that will come in this seminar. And he has been working with us. We're trying to bring him over to the dark side of mosquito-borne viruses, and it's been a real pleasure to work with him on that project. So with that, I will turn it over to Dan and just remind people, you can actually either use the chat or the Q&A function for your questions. We will address those at the end of the talk. Dan, take it away.
Great. Well, thank you so much for that introduction and again for the invitation to talk to you today. Let me just get my screen shared. Okay, so I'll be talking today about work that has really been kind of going on in the past 2-3 years or so, thinking about how can we kind of build better models to predict likely hosts of zoonotic pathogens. And so the work I'll talk about kind of combines a couple different modeling studies that get at different aspects of this question. And then also some more recent kind of empirical work that we've been doing in wild bats. So I don't have to kind of remind this audience, but we known for a while now that we think about all emerging infectious diseases in humans, that the majority of them come from non-human animals, and the majority of those species tend to be wildlife. And so a very basic kind of general question is just, kind of having this need to identify which wild species actually harbor these different zoonotic pathogens, including but not limited to viruses. The current pandemic we find ourselves in with SARS-CoV-2 really, I think, typifies the challenges that we face in identifying likely hosts of zoonotic viruses. And so with the exception of various cases of spillback of the virus into things like white-tailed deer and mink, we haven't really found the SARS-CoV-2 virus in any other kind of original wildlife host. We found some similar viruses like rat G13 in other species of bats, not this neotropical bat here, but a couple different species of horseshoe bats in Southeast Asia. And these viruses are about 96–97% similar to SARS-CoV-2, and molecular clock state divergence of those viruses from our current human virus to about 40 to 70 years ago. So the actual host that might have the human virus is still out there. It's still sort of unknown, despite lots and lots of sampling effort. And we also don't even know, other viruses that might be circulating. And so there's really this pressing need to identify the sources of not just known viruses like SARS-CoV-2, but also unknown viruses that might have zoonotic potential, say, any other beta coronavirus that might pose risks for human health. The problem, of course, is that there are many different host species. Just in mammals alone, we have thousands of species. And so we just can't logistically sample everything. This takes a lot of time, it takes a lot of funding, a lot of person power, and it's just not a task we can do. And so one of the solutions for how can we actually prioritize our sampling has been some work that I've been doing with colleagues at the Virina Consortium, thinking about how we can conceptualize any host pathogen association in a series of networks. And so this figure here shows a variety of different networks linking pathogens in kind of light blue to their different host species in dark blue. And so this just shows you kind of known associations in solid lines. And what we can then do once we have the structure of the host virus network is use various statistical methods to predict likely but unobserved associations. And so all the dashed lines here might be maybe unknown associations, but they're likely based on current patterns in our data. So all these models have very different structures, but in some sense, they all sort of boil down to the simple kind of linear model that we might all be accustomed to, where we have some response variable that we're predicting as a function of different characteristics, whether it's sort of network position or species traits. And so in this case, we are predicting something about host status, their ability to be infected or harbor a pathogen. And we're predicting that on the basis of these different species characteristics. And so this is a species level prediction problem. And this general approach with many different models has been really popular over the past decade or so. A lot of different groups using different models on different data sets to sort of identify likely hosts of various zoonotic pathogens. But despite the popularity of these approaches, there's still a lot of really big questions that remain about sort of how we do this kind of predictive task. And so one big question is sort of goes down to the structure of the model itself, right? We have countless kinds of machine learning models. Which of them actually work best in predicting host species? So this is a structural kind of problem. There's also an issue with what we're modeling, what the response variable is, what kinds of outcomes are actually most informative about host status. And then on the other side of that equation, we also have a question about the features we feed into these models. Which predictors could actually best improve our model performance? So today I'm going to walk through a couple different projects that get at these different questions in predictive modeling. For that first one, I'll talk about a long-term study we've been doing on bat hosts of beta coronaviruses. For the second question, I'll talk about some ongoing work we're doing with hantaviruses in rodent reservoir hosts. And then lastly, I'll also talk about some ongoing work that we're doing in neotropical bat systems, thinking about how we can characterize various within host or immunological features of bat species to improve predictive analyses. So thinking about this first question, beta coronaviruses are a really kind of nice group to focus on for this kind of study. So coronaviruses as a whole are very diverse. And beta coronaviruses include all of the high pathogenic coronaviruses. So SARS-CoV-1, MERS coronavirus, and our current SARS-CoV-2. And at least for the original SARS coronavirus, we do have evidence, right, that kind of bats are the ancestral host, at least, of these viruses. And so we know that coronavirus diversity in bats is very high, and bats seem to be the host of most ancestral beta coronaviruses. And so early in 2020, we worked with various group members in the Virina Consortium, computational scientists, virologists, other partners, including bat biologists, to sort of answer this general question in this particular bat beta coronavirus system. And we tried to boil down this big question into two more tractable questions. So first, if we have a bunch of different machine learning models, how well do these different models agree with one another in terms of their predictive capacity? And then not just making predictions, but also if we're actually able to validate our predictions with new data, that can help us get at this question of which models actually work best in predicting. This is something that is kind of like the holy grail of predictive modeling. We often make predictions, but then it's really hard to test them in real time. So to do that, we went onto GenBank initially, and we did a targeted scrape of any kind of bat coronavirus associations. We paired that up with available bat trait data for a total of around 1,100 bat species, so a little lower than the known diversity of bats. The bat species with coronavirus data are shown here in purple, so it's a relatively small fraction of bats, but it's fairly evenly distributed across the bat phylogeny. And we then built 8 different machine learning models, and these kind of fall into 2 main classes of models. At the top here, we have network-based models, which use information on the structure of the bat virus network, kind of who is connected to who through infection. And then we also use trait-based models, which use species characteristics to classify bats as those with a beta coronavirus association from all other bat species. And then because each of these 8 models predict something slightly different, we ranked our predictions and then made this proportional rank measure on the x-axis. And so a lower proportional rank means you are more likely to have a beta coronavirus. And so what I'm showing you here is the kind of the raw data. So the ones are known beta coronavirus associations. The zeros are unknowns. And then these fitted curves are just from a logistic regression. And so what you can see is that there's a lot of variation within the network models, where network model 2 performs really well at discriminating known and unknown bat hosts. Network 4 does okay. Network 1 and network 3, not so much. If we go down to the trait-based models, we find that in general, they seem to perform a bit better, especially hybrid 1, trait-based 1 and 2. They generally do a bit better job differentiating those known from unknown hosts. And we then took those 8 models and we put them into a total multi-model ensemble, which, as you can see here, sort of generally captured most of the good predicted performance and beat out all but sort of 2 of the individual submodels. So this is kind of our ensemble model. Right, and then one of our main questions was how well do these predictions between models agree with each other? And so to do that, we looked at the correlation between each individual model's predictions with every other model, and that's shown here. And so any of the darker red colors are strong agreement, blue colors are strong disagreement. What we generally find is that if you look at just the network models or just the trait-based models, there's a lot of agreement kind of within model taxa. But if you start looking at how well the trait-based models relate to the network models, there's really not much strong agreement at all. So there's more agreement within rather than between different groups of machine learning model types. We then use these models to make kind of a prioritized list of likely hosts based on these predictions. And so with a couple different cutoffs, we identified an initial 371 likely hosts of beta coronaviruses. And these maps just show you the distribution of predicted hosts for each of these model types. You can also compare that to the distribution of observed hosts and kind of get a sense of where there is mismatch or not mismatch. So for example, right, there's kind of a higher density than you might expect based on the data in North America and South America. We kind of recapitulate this known hotspot of beta coronavirus diversity in Southeast Asia, but our models kind of maybe get away from some bias that you see in the data in Europe, especially. We don't see as strong of a hotspot in those areas per se. And so it's all well and good to sort of make these initial predictions. And then we wanted to then take one step forward and actually sort of systematically start to test them. And so to do that, between 2020 and 2021, we systematically tracked any known and new beta coronavirus associations. And so this is all documented on the CREATE-NEO website. We have that beta coronavirus page you can visit to sort of track this documentation process. We did this a couple different ways. We did standardized literature searches through a sub-project we have called DataCove, compiling coronavirus prevalence across bats globally. We went back to GenBank and re-downloaded any new record, of which there are many, for beta coronaviruses during that whole year. And then one of our collaborators, Laura Bergner at University of Glasgow, also did some mining through available metagenomic and transcriptomic data for any signatures of beta coronavirus infection. And what this did was give us 47 new hosts within just the span of that 1 year of new beta coronaviruses. And we then compared basically how well did our model do at predicting these new hosts. The ensemble as a whole got 36 of those 47. So we're doing pretty well, 76%. We can then break that down a little bit more into individual submodels. And so one way to think about that is that we can compare the prevalence of predicted positives from any one model against how many hosts they correctly identified. And so you can think about this like a receiver operator characteristic curve, where anything along this one-to-one line is kind of the 50-50 shot. So if a model is working at random, it's going to predict 50% hosts, and it'll identify 50% of those hosts. And so what we really want is for the models to be in the upper left-hand corner of this diagram. What we found, if you think back to the earlier slide, was again that the network models overall did pretty poorly. So all the red lines here are really hugging that one-to-one line, whereas the trait-based model and the ensemble, the blue and the black lines, these all sort of are starting to go up into that upper left-right corner, top left corner, kind of showing an overall improved performance. And we can summarize that a bit better just with the overall sort of performance values. And so in doing that, again, you can see the network models are kind of hugging the one-to-one line. They're working kind of about random, or in some cases like network 2, they're working worse than random. Whereas the trait-based models on the ensemble are doing much better than expected at correctly getting these known hosts. So there's a couple kind of big take homes we can get from just the modeling side of this. So first off, we kind of see a decent amount of agreement, but also disagreement between different kinds of models and how they predict host species. But when you look at the ensemble as a whole, this can actually be a really useful tool to buffer against that variation in model quality. Our ensemble generally performed really well. It kind of took the good parts of the models and left behind maybe some of the suboptimal parts. And maybe kind of most importantly here, we saw that the trait-based models did a really good job compared to other model structures. And what that really tells us is that if you want to make good predictions about a host species, and this is almost a intuitive result, you have to have detailed data on the ecology and evolution of those species. And so it really emphasizes the importance of understanding the natural history of the species you are trying to analyze in making good predictions. So that's kind of the modeling take homes of this study. What about the kind of pragmatic, beta coronavirus surveillance take homes? So we took those models and we re-updated everything with those 47 new bat hosts. We reran our ensemble, and this gave us a 412 new likely hosts of beta coronaviruses using basically all available bat coronavirus data. This map just shows you the overlapped distributions of all those predicted species. And so what we see here is kind of a geographic shift in those hotspots. We still have a high concentration in Central Africa, but we also see some of the maybe riskier areas move from mainland China down to the Malay archipelago. We also looked at taxonomic patterns in these predictions. And so in the bat phylogeny here, every line is sort of scaled by that rank prediction. And we then used another machine learning tool to identify kind of clade-based clusters of risky hosts. And so the clades in red show you those that have a really high probability of having undiscovered beta coronaviruses. So unsurprisingly, the rhinolophid bats pop out, and this is where we've kind of found a lot of the closely related beta coronaviruses in bats so far. We also found some subclades of pteropid bats, including bats in the genus Rosetus. And then kind of surprisingly, the molluskids as a whole were in blue here, kind of a low risk clade, but we found sort of the old world molluskids, especially the genera mops, kind of stand out as a group of bats that might be especially likely to have undiscovered beta coronaviruses. And so we can take these predictions and kind of take them and then do model guided sampling, both to characterize what these new beta coronaviruses might be and try to understand their dynamics in nature a bit more. And so I thought I would just kind of briefly talk about two projects we've been doing to try to do this empirically. One is using museum collections to further test these predictions, initially focused on the predicted species in the genus Rhinolophus. This is work led by Adam Ferguson at the Field Museum of Natural History. And as some really kind of cool preliminary work we found, we recently had a new beta coronavirus pop up in Rhinolophus fumigatus in Angola. This definitely falls within the beta coronavirus group. So it's a really kind of nice example of we've prioritized these species, and we actually find a new kind of beta coronavirus in some of these groups. We're also doing some similar work at University of Oklahoma in my lab, working primarily in the Americas, studying both phyllostomid bats and molluskids predicted by our model. So in Belize, we work with the Laminae Field Research Center and a number of other collaborators to sample some of these predicted hosts like Sternauer-parvidens, Glossophagus orsina, Molossus rufus. And then in Oklahoma itself, we're also sampling some of our migratory bats predicted to be beta coronavirus hosts like Tederida or Brasiliensis. And so hopefully in the next year or so, we'll have kind of some interesting updates on that front on how we're doing testing these predictions in real time, and also understanding how these viruses circulate and persist in their bat hosts. So going back to our really general question here, if we think about kind of this first task of which models work best, I think we have pretty convincing results that it's really trait-based models that we should be using if we're trying to predict likely but unsampled host species. And so that's very much, again, kind of finding this is the kind of model that we should be doing. As a follow-up, then we can think about what are the ways that we can improve the kinds of data that we're feeding into these models, both on the response variable side and on the predictor matrix side. And so thinking about both response variables and predictor variables, we've been really thinking a lot about kind of within host data. And so in a paper we wrote 2 years ago now, we thought about what are the different processes that are occurring within the host once they're exposed to a virus, right? So you have susceptibility, the pathogen then has to replicate, and then the pathogen then has to be shed or survive in the environment for long enough for a new host to pick it up. And especially in the ecological immunology literature, we kind of encompass all these terms in the really broad characterization of host competence, or the ability of a host to actually transmit a new infection to another host. And so if we think about that kind of simple base model that we have for encapsulating host virus interactions, there's a couple different ways that within host data can enter into this equation. The most ideal version is that we actually can do experimental infections, right? We can take animals, infect them, and see how well they actually replicate the virus, pass it on to new hosts. And this could be a really nice way to both generate a new response variable and also systematically test predictions. And there's been kind of a handful of case studies that have done this in a really nice way. For example, a recent study predicted that Rosetta's is a good host for Nipah virus. And then some work by Steph Seifert at Washington State convincingly showed that you actually can't infect the species with Nipah virus at all. So it's a good way of kind of rejecting that prediction. And likewise, there was a recent study that challenged big brown bats with SARS-CoV-2 and also found that despite lots and lots of different methods, you can't infect big browns with our current coronavirus at all. So another way of kind of canceling out potential predictions there. We also know, though, that experimental infections are costly and challenging and hard to do at a large enough scale for predictive models. And so there's at least two other ways that we can start to bring in some of this within host data into our models. So on the one hand, we can try to model competence directly through some proxies that are maybe more feasible in the field. Things like actually getting live virus from a host might be one way that's a bit easier than doing experimental infections per se, actually showing that that animal is replicating virus, is actually shedding virus. We can also think about the predictor matrix. And so we can take, if we could generate enough immunological features across a large enough number of species, those could then be fed into our predictor matrix as maybe more biologically relevant features of the host virus interaction. And so these two sort of final projects are just going to focus on the one hand, how we model the response variable in rodents, and then on the other hand, how we might start to develop some better within host predictors in wild bat systems. And for that project on sort of rodents, this was a collaboration with Chris Forbes and Nathaniel Mill at University of Arkansas. And we focused on rodent hantaviruses in the Western Hemisphere, in part because this team had already put out a really comprehensive systematic review of these viruses, which cause a number of different human diseases in both North and South America. And again, in this context, specifically, shedding live virus provides a good kind of field approximation of competence. You're able to find that the rodent host is actually susceptible to the virus, is actually replicating the virus, and then is actually causing new transmission events when it sheds. So with Nathaniel and Chris and Colin Carlson, we gathered data on about 600 different rodent species in the Americas, and whether or not they are either actively infected through PCR or whether they've had successful live virus isolation, indicating sort of this competence variable. And much like our coronavirus study, most of the species in this data set are zeros. We have a small number of PCR positive rodent species, an even smaller number of live virus positive species. And we then took the top model from our beta coronavirus study. So in this case, this is a boosted regression tree, a very popular trait-based modeling approach. And we ran a variety of different models with a bunch of different ecological traits, using PCR as the response variable, and then virus isolation as the other response variable. And we then measured performance as area under the receiver operator characteristic curve. This just shows you the data across 100 different partitions of training and test data, so to make sure that we're actually testing our models out of sample. We found that in general, right, all of these models are performing really well. The lowest AUC we find is 0.8, which by viral ecology standards is still really good. But we definitely see that models based on virus isolation do significantly outperform kind of PCR only models. And if you standardize this into a normal measure of effect size, we are seeing a very strong effect by standards like Cohen's d. So it's safe to say that using virus isolation as the response dramatically improves model performance. Although if you're interested in kind of the inference from these models, we actually find that inference remains pretty similar. Both models identify the same, by and large, the same features as predictive of host status, with a few exceptions in terms of taxonomic patterns here. So it might be more interesting to look at, not only how do these models vary in terms of their performance, but how do the predictions themselves vary? And so to do that, we plotted the predictions from the PCR models against the predictions from the virus isolation models. And we did find some relationship between these. If you care about the stats, it is significant, but again, the effect size here is really small. We're seeing a very small correlation coefficient. And there's kind of some interesting stuff you can tease out of this figure. So on the one hand, we have a number of species that have very low predictions if you were basing this just on PCR, but that have fairly high predictions based on virus isolation. So these are species that if you were trying to prioritize surveillance based just on PCR, you might not ever think to actually look at these species because they're all the way down on the left side of that graph. On the other hand, we have a number of species that have very high predictions based on PCR models, but very low predictions based on virus isolation models. And so these might be sort of false positives from a surveillance point of view, where we might go out and sample them based on models based on PCR data, but we're unlikely to actually maybe see kind of true sort of reservoir hosts, since the odds of them being capable of actually shedding live virus are so low. So the correlations are weakly predicted, and we might often get a lot of false positives if we're basing our models just on PCR data rather than trying to get at virus isolation or competence per se. And lastly, we can also look at how these predictions also tell us something different about the geography of hantavirus risk. And so here we're just overlaying all the known PCR positive species and the virus isolation positive species, and then also how those contrast with the predictions from our models. And so what you can see if you look at the PCR positive predictions, we kind of have this hotspot on the eastern coast of Brazil, in parts of Central America. And really those geographies kind of shift when we start looking at predictions based just on virus isolation, where now we're maybe looking more at the northeastern US, or sort of the western area of South America, including sort of Peru and Venezuela. So if we go back to the general issue of which outcomes are most informative about host status in these models, it's safe to say that modeling host competence not only improves our predictive capacity, but it gives us predictions that are actually fairly different from what we might get based on just models about infection alone. And so lastly, I just want to talk about some more recent kind of new work that we've been doing on thinking about the other side of that model structure in terms of how might we generate more within host or immunological data that we could eventually feed into larger models. So this work is based on a long-term field study I've been doing in northern Belize with folks like Brock Fenton, Nancy Simmons, the Lamanai Field Research Center outside of the Lamanai Archaeological Reserve. And since about 2014, we've been doing rabies virus surveillance in a longitudinal study in wild vampire bats, which we know are kind of the dominant reservoir host of rabies in Latin America. And so back in 2017 or 2018, I got in touch with Ben Neely at NIST, who's a big proteomics expert, who was interested in kind of building a large comparative database of serum proteomes across different mammals. This is a really cool initiative, and if you look on the NIST website, you can get more insights into it. Ben wrote a really nice review paper a few years ago just on sort of applying proteomics to non-model taxa. And one of the main benefits of proteomics in this case is that we're getting not just the proteins circulating in blood, but also the proteins circulating that are kind of leached from other tissues like liver and kidney, for example. And so it's a very sort of holistic way to look at proteins in an organism. And then the methods that we've been using only require about 2 microliters of sera. So something that's actually very feasible for even some of the smallest bat species that we might sample, maybe in contrast to having to use whole blood or even organs for other sort of omics techniques. And so in last year, we did a kind of pilot analysis of characterizing the serum proteome of wild vampire bats in Belize. And this is kind of one of the summary figures from that paper. So we identified about a little under 400 different proteins in sera, spanning about 5 orders of magnitude. The plot on the lower left just shows you the overall abundance of some of the top proteins. And then just for the sake of immunological interest, we kind of highlighted any kind of complement-based proteins in orange on that rank abundance chart. And so we find that complement proteins are very abundant in bats, kind of span the full range of various proteins in plasma. And so you could think about, further in the future, that if you were trying to build a big global host virus predictive task, that you might be able to use these sorts of data compiled in enough species as predictors. So one nice application to this, colleagues at Virena recently built this really large data set of host virus associations across not just mammals, but also birds, fish, what have you, called the Virion project. So I encourage you to look into that if you're interested in kind of really large scale prediction. And so, right, if we could characterize the proteomes of enough bats, we could take all these different protein abundances as features and feed them into that kind of model, and that might end up improving predictive performance. That said, right, if we are thinking about a really focused trait-based model, like predicting bat hosts of coronaviruses, it might kind of behoove us to focus not just on the entire proteome of 400 proteins, but maybe try to narrow in on specific proteins that we know have some immunological or pathological relevance to what's happening in the bats. So recently we started doing that in the vampire bat system based on some preliminary work with Ryan Relic at Indiana University. We've characterized a couple alpha coronaviruses that do seem to be both fairly novel. So the phylogeny here just shows you 4 positive swab samples that we have that essentially cluster out a bit more closely with known human alpha coronaviruses than other neotropical bat alpha coronaviruses. And they're also prevalent enough that we can actually look at some differential abundance. And so we had about 4 of 20 swabs test positive from a really early sample. We then took paired sera from all those swabs and submitted them for another big proteomic analysis. So the plot on the left is just showing you again the rank abundance of our proteins, and we here identified just under 600 proteins this time, again spanning about 5 orders of magnitude. And on the right, we then have sort of the scatter plot of the mean abundance in uninfected bats against infected bats. And so immediately clear is that we don't find much evidence for differential abundance based on alpha coronavirus infection. And so we tried looking at this a little bit differently. We then used receiver operator characteristic curves, so a basic classification task, to identify about 30 proteins that have decent classifier ability, defined as a AUC above 0.8. And so those are shown in gray here, and then the really good classifiers are those with AUC above 0.9, and those are shown here in black. And so some of these are familiar proteins like complement proteins. Others are some that have some immunological relevance. Others we might not classically think about as immunological proteins, but they're still standing out here as likely biomarkers of infection. And here we can just kind of get more insight into whether those are kind of positive or negative predictors of infection status. And so things like C4A involving complement synthesis are generally kind of downregulated in our infected bats, whereas others like GST01 tend to be sort of upregulated in infected bats. If we take those 30 biomarkers and just look at basic descriptive patterns of abundance, it seems like they do a pretty decent job separating out the infected and uninfected bats, with the exception of this one horrible outlier infected bat kind of in the middle of the plot here. And we also did an enrichment analysis to try to identify just up and down regulated immune pathways in the system. And so some kind of clearly downregulated processes in coronavirus infected bats included a downregulation of complement and the humoral immune response, whereas we saw an upregulation of primarily a cell-mediated immune response, especially involving granulocytes and neutrophils in particular. And so again, going back to sort of big scale predictive modeling, you can imagine that if you were building a coronavirus-specific predictive model, this might give you some good insights into particular proteins you could target to characterize across a range of bat species that actually have some insights into coronavirus pathogenesis, right? So those, if we did those with enough species, and again, we're dealing with 2 microliters of sera here, so it's actually feasible, right? Those could be built in as predictors into some kind of larger matrix in this predictive task. So that's something that we're really interested in moving forward with. It seems like some of these protein biomarkers might actually give us some sort of meaningful, biologically meaningful viral features. Most of what we're doing, again, is sort of larger scale ecological or geographic predictors. And so this might actually tell us something about not just exposure to a virus, but the sort of pathogenesis or immune response to the virus itself. And thinking more generally about kind of the different projects here, I think these are all really concrete steps in how we can improve and optimize our approaches to predictive science in kind of viral ecology. And I just wanted to kind of end on a note that these improvements to modeling don't just inform kind of academic practice, but ultimately they are kind of feeding back into really what we are sampling, and in even some cases, sort of how we target interventions. So some colleagues and I wrote at the start of the COVID pandemic about some concerns about the ways that that virus relationships might be leveraged towards negative perceptions of bats, or even things like culling particular species. And so the ways we build these models and what species they're prioritizing for surveillance or even for interventions might really matter for downstream sampling campaigns or approaches to actually try to limit that virus spillover. And so with that, I just want to thank a long list of collaborators, funding sources, and then of course, you all for your time and the invitation to come talk today. And that is it for me.
Thank you, Dan, for an excellent talk. We have a couple questions. I'll start with the one in the Q&A from Artem Babyan. Really cool talk, thanks. How specific do you think the proteomic pathway signals are to coronavirus infection versus other RNA virus infections?
Yeah, that's a great question. And we're dealing with, again, a really small sample here. This is like 19 individual bats, 4 of which are positive. So we're dealing with a very imbalanced sample. But again, part of the benefit of this approach is that the proteomes are there, and we have RNA extracted from these samples. So feasibly, right, we could go back and test those same swabs for other RNA viruses, look at similar patterns of differential abundance, different enrichment pathways. And so a lot of the work we're thinking about moving forward is obviously using larger sample sizes, working not just in vampire bats, but across a variety of other neotropical and North American bat species, and really seeing how consistent some of these either differential abundance patterns are, or how conserved some of these pathways are across larger clades of bats as a whole. So it's definitely something that we're thinking about. And I think applying that to a larger suite of viruses would, of course, be interesting, assuming we can find enough positives across kind of the breadth of bats we're thinking about.
Great. Question from Rania Molchandani. Thanks for the really interesting presentation. Apologize for what might be a silly question, as I have limited training in modeling. In your first section, where you compare the performance of the different models as well as the ensemble of all of the models, did you consider only combining the higher performing, say the 2 to 3 with the highest R value, rather than all the models as your ensemble? Thanks.
Yeah, that is also a great question. So initially we left all the models as is so that we kind of do this post hoc comparison moving forward. One step that I didn't talk about, so after we collected all these new 47 new hosts, we reran all the models, we did see an overall increase in performance. So the R-squareds for each individual model bumped up pretty decently. The ensemble went from, I think, 0.42 to about 0.6 something. So a pretty big increase in predictive performance. And then when we built our final ensemble that those predictive maps and phylogenies were based on, we did do kind of some weighting scheme for basically how successful was each model at predicting those new hosts, and then weighting the models by, how well did they do. And so our final ensemble kind of took variation in model performance into account, just so we're not treating the lesser models the same as the optimal models. So yeah, definitely something that we thought about, and it's reflected in those final predictions.
And then I think you may have already answered the second part of the question, but what is the overall goal of the improved approaches for more targeted enhanced surveillance of wildlife? I think you said yes, that is the goal.
Yes, yeah. I mean, so there's the one end of how we can just learn to build a better model, but on the other end, right, how can we, if we're thinking about bats and we have 1,420 something, I think is the most recent count, right? How do we whittle that down to a manageable number of species? And that's going to be based both on geography, which bat species are present, and then all the logistical costs that go along with doing all the field work and lab work. And so, taking that big list and using these models, even narrowing it down from 1,400 to 400 is a pretty dramatic reduction in the amount of time and cost it might take to do that kind of surveillance.
And then you have a question from Nick Komar. With vector-borne viruses, we can use mosquito blood meal analyses to build species networks. And I feel this question because we do this too. Do you think these network models would be more useful than using these vector-based approaches, I guess?
Yeah, so there's definitely been some really neat work from the Varuna Consortium in expanding the kind of host virus networks, which are, you have two things you're connected to a larger network of host vectors and viruses. And so there's a preprint out led by Tad Dallas that you could look into that considers that more complicated network structure. And definitely for mosquito-borne or any kind of vector-borne virus, you do have that additional complexity of the vector built into it. And so in some sense, network models might work better, but at the same time, the trait-based models can also consider not just traits of the host, but also traits of the virus, traits of the vector. And so this preprint by Tad does a really nice job in starting to try to tease apart all those different contributions to the host vector virus relationship and make predictions based on those.
And Dr. Komar had a follow-up question. What kind of traits are used in the trait-based predictive modeling for hosts?
Yes, and so most of this to date is used on things that we can get in a standardized fashion across lots of species. And so in the beta coronavirus and the hantavirus project, we mostly used data available in Pantheria, which is data on geographic distributions, body size, some life history data, dietary data, population density, things that are relatively standardized, although acknowledging there are a lot of big data gaps in those databases as well. And one of the critiques of those is that even though they're really more accessible, right, a lot of those are probably things that are telling us more about host exposure to a pathogen. Or maybe they're proxies, like body mass might be a good proxy for some actual within host process about, once an animal is infected, how well does it deal with infection? So the work kind of that we're starting to do with bats and the proteomes in particular is kind of an effort to build on those ecological predictors and try to add more immunological data into these models. So I think that's really kind of what's missing right now. And understandably, it's really hard to get that data across lots of different species. But I think that's kind of where we're moving.
Great. I have a question, and then I'm going to read Dr. Terrell's question. So my question was regarding those 4 bats that were positive for alpha coronaviruses and the 16 who weren't. Were there other differences between the bats that might have explained those proteomic differences? Were the infected bats younger, different sex, anything like that?
Yeah, and I think this is one of the challenges with the small sample size right now. So they're all from the same site, they're actually all captured on the same night, emerging from their roost. And so we don't really have any kind of temporal or spatial issues to deal with. If I'm remembering right, the majority are males, but there are some females in there. There are some females that are lactating or pregnant. And so, again, another thing we want to look at when we start doing this more systematically across more bat species and larger sample sizes would be, in that that heat map that I showed, right, you could also pile on, the sex of the bat, the reproductive status of the bat, the age of the bat, and sort of see how well that might also contribute to those proteomic differences. I'm sure there is plenty going on there, especially with reproduction. In the free-tailed bats, we're really interested in the contribution of migration to those kind of differences. So, kind of trying to get at the contribution of some of the individual level factors, and then also the species level characteristics, I think will be a really exciting area to go into.
Great. Michael Terrell asks, thanks for the interesting presentation. I'm curious about the comparison between virus isolation and PCR data. It is much easier to get large sample sizes with PCR, but as you found, PCR can often detect pieces of non-replicating RNA that result in false positives.
Yeah. So one of the things that study is available as a preprint. It's in revision right now, but one of the challenges, right, is that you can't do virus isolation for everything. And so kind of that approach is probably best suited to those systems where it's at least feasible. If you think about something like some of the hendipoviruses, right, like in some cases, those have never been isolated from a certain bat host. And so that wouldn't be a good candidate for that kind of system, but other viruses where you might have at least some fraction of known hosts with isolation data would be more amenable to that sort of approach. We've thought about similar things with some tick-borne pathogens like Borrelia, where you can have transmission from a host to a naive larval tick. And so in that case, finding a Borrelia positive larvae on a bird or a rodent is kind of another approximation of competence. And so those are other systems where this kind of approach might be beneficial, but it definitely doesn't apply across the board. And so I think our suggestion would be in cases where you can't do that kind of infection versus competent comparison, that maybe switching and trying to think about what are the immunological features that you could put into the predictor matrix might be a better way to try to at least capture some of that within host interaction.
A very quick question from Tanya Roma. Is the Becker and LEI available as a preprint?
It will be in a week or two.
Yeah, in a minute. Okay.
We're getting our gem making sessions up there. So as soon as that's done, we'll be posting that as a preprint. But yeah, it's coming.
And then a question from Christian Cabrera. Do you have some advice for researchers like me in Colombia to begin to access the virus diversity of mammals?
Yeah, but I think looking into the Virion database that the Virina group has put together is a really good first start. There's a really nice kind of tutorial that's up on GitHub, introduces you to the database, how to access this really enormous host virus data set. And there's also a preprint up for that project that walks through a couple different examples. And so that might be a good place to get started on following along with some of that code and kind of how to access the database and how to manipulate it, which things to filter out, that sort of process.
Okay, great. Sarah Woodson says, great talk. Have you tried to incorporate any climate data projections, 5-10-year increments, to see if there's any projection on how this targeted sampling approach may shift over time? Yeah,
that's a great question. So some of the work by Colin Carlson and Greg Albery here listed on the slide. There's also a preprint up on sort of looking at climate shifts and viral sharing across mammals, which you should definitely look into. Another interesting kind of approach here is, taking some of these predicted hosts and just like you're saying, ask how their distributions will change under different climate scenarios. It's something that we've thought about doing in some of our Borrelia work. It's something that you could probably apply to most of these systems. It would just take, downloading the shape files of the predicted hosts, running some of these different climate models, and then asking how their distributions will change. So I think that's a really promising avenue for asking, not just what should we prioritize now, but what should we maybe prioritize 5, 10, 15, 20 years from now as well.
Although I'd like to just take the moment to cheerlead better distribution maps for a lot of these host species, because we constantly run into the fact that we get this sort of range outline that is not terribly useful when you want to make those kinds of more fine scale predictions.
Right. And I think one other take home from these different projects is, just data quality really matters, right? Whether it's kind of what you're feeding in as the response or what you're feeding in as the predictor, but goes the same for climate modeling as well.
All right, one last question. Michael Terrell says a quick follow-up. Yes, having an immune response does indicate a prior infection. However, for some viruses, the viremia level never reaches a level where that vertebrate host can serve as a source of infection for another host. To prove that a particular host is involved in the transmission cycle, you need to show that the virus can replicate to a level where it can be transmitted to another host.
Yes, and that's a really good point. And the kind of isolation data that we use in that hantavirus model, right, doesn't take into account viremia, for example. I mean, it's something that, if you could have that data, that could be your response variable instead. Or you could take the viremia and apply your binary cutoff or whatever that transmission cutoff is, and then that could be your new response variable. So it's definitely something that isn't incorporated into that hantavirus example, but in other systems where you would have kind of those known cutoffs, you could apply that in the same fashion to again indicate which species can now generate enough virus to actually cause transmission versus which ones still aren't generating enough and should still be considered a zero.
All right, well, that's all the questions that I see, and it was a great session, great talk. So thank you, everybody, and please come back on February 8th to hear Dr. Sandra Lopez Verges talk. She hasn't given us her title yet, but I guarantee you there will be arthropod-borne viruses in it somewhere, and her title will be posted shortly. Thank you, Dan. That was great. You had a great audience. There were 130 people on at one point. So
yeah. Thanks again for the invite and all the really good questions.
Yeah, no, they were all great questions. It was a very lively session. People are coming back from their holiday break all energized.
I got the good
science again.
Yeah.
Not hang out with my family. And you're getting lots of things on. I don't know if you can see the chat or not, but
yeah,
great. Trickle off. Make sure there's no last comments. All right, Ashley, I think we're good to shut down then.